What if everything that makes your deepest fears, your most soaring ambitions, even those little quirks that make you smile, isn't just fate or how you were raised? What if it's something written, like a hidden language right inside your cells? What if your destiny, your health, your identity are all tangled up with this silent, powerful code? Today, we're starting an incredible journey, really getting into Siddhartha Mukherjee's amazing book, The Gene, An Intimate History. And this isn't just, a science textbook. It's this huge story that weaves together his own family history with the massive scientific quest to figure out heredity. The story of discovery, yeah, but also controversy and what it all means for being human.
Right, and our mission here is really to unpack the big themes, the breakthroughs in science, and the honestly profound implications of getting a handle on our genetic code. We'll look at how this book mixes the personal, like dealing with illness in a family, with these huge universal scientific discoveries.
Exactly. And if you find yourself getting drawn into this kind of deep thinking, pausing to chew on complex ideas, you might also really like the Mindful Moments YouTube channel. It's a great place for that kind of thoughtful content. Okay, so let's start way back. Looking at the very first print tries to figure out what makes us tick. For, millennia, people puzzled over heredity. Ancient thinkers actually had some surprisingly, radical ideas. Thinking about someone like Aristotle, what were his really visionary insights, especially about what gets passed down?
Yeah, Aristotle ages ago had this insight that feels astonishingly modern. He sensed that heredity wasn't about physical stuff, but information, pure information. He called the male contribution a principle of movement, but he meant instruction, information. It's incredibly close to how we think about code now.
Wow, instruction, not matter.
Exactly. He argued that what passed from man to woman wasn't material, but a message. Like a carpenter giving plans for a building, not the wood itself. The semen carried instructions. It's like a fundamental aha moment, conceptually mirroring the genetic code centuries before anyone knew about DNA. He even noticed traits skipping generations, like mentioning an Ethiopian woman whose granddaughter has similar features.
That's pretty profound for back then. Okay, right. So jumping forward, leaving the ancient world, the 19th century throws up its own big challenge.
Yeah.
Sir John Herschel called it the mystery of mysteries. How did all the different species come about? Charles Darwin, on the Beagle voyage, he collected, Mukherjee calls it, cargoes of apparent rubbish.
Right, plants, animal skins, these enormous fossils like the jaw of a megatherium, that giant sloth.
Yeah, but he wasn't just, collecting things. He was looking for something deeper, patterns.
And that's the key thing. Darwin's genius wasn't just observing, it was seeking the underlying systems. He wasn't just classifying. He wanted the mechanism of change. And the clue came from, a kind of unexpected place, Thomas Malthus's essay on population.
The economics guy.
Yeah, the controversial idea that populations always struggle against limited resources. That economic theory gave Darwin, and amazingly Alfred Russel Wallace too, the missing piece. It led them both independently to natural selection.
And Wallace. That parallel discovery is just mind-blowing, separated by oceans, different intellectual paths, but they land on the exact same mechanism. It really underlined that evolution needed both things. Heredity had to have constancy, so traits get passed on, but also inconstancy, mutation, for new variations to pop up.
Which brings up a really important question.
Right.
Okay, variation happens, but how exactly does it get passed down? August Weismann gave us a huge piece of the puzzle with his germplasm theory. He argued that hereditary information is only in the sperm and egg cells. So acquired traits, like the classic example, a giraffe stretching its neck its whole life to reach higher leaves that stretching cannot get into the germ cells. Doesn't matter how much the ancestor stretched, it couldn't pass that specific acquired length to its babies. This really helps separate heredity from individual life experience.
Okay, so Weismann helped rule out what doesn't get inherited. Who finally cracked the actual rules? That brings us to Gregor Mendel, right? Almost a tragic story. His paper on pea plants, 1865, totally groundbreaking.
And totally ignored for decades.
Yeah.
Then in 1900, Hugo de Vries finds it. Mukherjee describes that feeling, that chilling déjà vu of realizing someone got there first.
Oof.
But what de Vries figured out, even if Mendel was first, was still critical. He saw these spontaneous mutants. He studied this plant Onothera with giant leaves and realized these were Darwin's missing variations. These random mutations created the raw material that natural selection could then work on. He had that great line, "A chicken was merely an egg's way of making a better egg."
Meaning life isn't just about survival.
Right. It's about the genes, the information, perpetually improving itself, evolving. a really profound shift in thinking about what life is fundamentally for.
But, with great scientific power,
Mm.
often comes pretty big ethical problems. As we started understanding genetics more, this really dark chapter opened up. Francis Galton, Darwin's cousin, he coins the term eugenics. Started off sounding okay, like human emancipation,
Mm-hmm.
but it morphed into a macabre form of social and political control. It's easy to condemn it now, but what made it seem logical, even like progressive to smart people back then? How do they justify it?
It's a really chilling lesson in how science can get twisted. People genuinely thought they could improve humanity, but it quickly turned into creating hierarchies. H.G. Wells, in The Time Machine, he imagined a future where desirable traits were pushed so far, even suggesting selective elimination of the weak. And this wasn't just science fiction.
Right, like the Buck v. Bell case.
Exactly. The U.S. Supreme Court, 1927, Buck v. Bell, Carrie Buck,
Mm-hmm.
legalized compulsory sterilization. Justice Oliver Wendell Holmes Jr.'s horrific line, 3 generations of imbeciles are enough. It just shows the dehumanizing potential, the idea that some lives were just unworthy.
But some key geneticists pushed back. Herman Miller, who was initially into eugenics, he realized it was fundamentally flawed. He argued it just became corrupt without real equality. He saw people like Carrie Buck not as genetic imbeciles, but as victims of their social lot. He saw that without equality, eugenics was just a tool for control.
And the significance there is that Nazism took this to the absolute extreme. They called it applied biology. Fritz Lenz used that term. The whole concept of Lebensentwürdigkeitsleben, lives unworthy of living, led straight to the genetic final solution. It also caused this huge brain drain from Germany. Brilliant scientists like Einstein, Hans Krebs, Max Delbrück, they had to flee. The situation was just morally impossible.
A really dark period, but somehow the scientific search for the gene itself kept going. The discovery of DNA structure was coming. It's almost funny now, but early on, biochemists like Phoebus Levine thought DNA was a stupid molecule.
Yeah, like too simple. Just a repetitive polymer couldn't possibly carry complex genetic messages. They figured proteins must be the key.
How wrong can you be?
Completely. Oswald Avery's experiment in 1944, just painstakingly brilliant work distilling this transforming principle in bacteria. He proved DNA was the material substance of the gene, the actual cloth from which genes were cut. And the irony, this huge discovery, finding the blueprint of life, happened right as the Nazi exterminations were reaching their peak.
Yeah.
Which shows how scientific truth pushes forward, even in the absolute darkest times.
So that set the stage for Watson, Crick, and Wilkins. Their work, described as having this element of whimsy and intuition, building models, led to the double helix. That image, it wasn't just science, right? It captured the whole feeling of the 1950s, that optimism, that wonder. It promised we could rewrite our nature. What did finding that shape actually tell us about how genes work?
It was like finding the Rosetta Stone for life. It changed the gene from this fuzzy idea of a message into a specific readable code. For the first time, you could physically see how instructions were written. You understood how changing just one letter, one base pair, could encode a huge change in someone's life, like with sickle cell disease. Mukherjee calls it a Rube Goldberg disease, one tiny chemical tweak causing this whole cascade of awful effects. The elegance of the structure explaining the function, it was stunning.
Okay, so double helix understood. What's the next big leap in thinking about genetics, especially with health and disease?
Well, we move into the modern era, and this idea emerges, articulated by Paul Berg, that maybe all human disease is genetic in some way. We started understanding genetic diseases with way more subtlety. We could distinguish between monogenic diseases caused by one gene, like Marfan syndrome, and the more complex polygenic ones, like diabetes or schizophrenia. Those involve multiple genes plus environmental factors.
Right, there's not just genes, he's destiny.
Exactly. The formula becomes something like genotype plus environment plus triggers plus chance phenotype. Your genes plus your life plus random events equals the outcome. Take the BRCA1 gene mutation. It gives a woman maybe an 80% lifetime risk of breast cancer, a huge risk, but it's not 100%. Because of incomplete penetrance, it doesn't always fully show up. Chance still plays a part.
That more nuanced view leads us into some tricky ethical areas, doesn't it? All the new capabilities like prenatal testing, selective abortion, some people call it nugenics or neo-eugenics. How is this different from the old eugenics, and what are the dilemmas we're still wrestling with?
Well, the key difference is supposed to be individual choice. It's not the state forcing things like in the past. But critics argue, correctly I think, that the line between wellness and illness is blurry. It's a spectrum, continuous kingdoms, as Mukherjee puts it. What one person sees as a defect to be screened out, another might not. It's subjective.
Yeah.
And you see the weight of this in Nancy Wexler's story. Her personal connection to Huntington's disease, this awful waiting game if you carry the gene. Her work finding that cluster of cases in Venezuela was absolutely crucial for mapping the Huntington's gene. It shows the power of genetics to shed light on terrible conditions, but also the really tough choices that knowledge forces on people.
And genetic diversity also gave us tools to understand where we all came from, right? Things like the molecular clock.
Yeah, looking at mutations that build up over time, especially in mitochondrial DNA, which you only inherit from your mother. It helps trace human relatedness, migration patterns thousands of years back, and this has huge implications for the whole idea of race. Because when you actually look at the genomes, the amount of variation between human populations is strikingly low, especially compared to, say, chimpanzees. We share 96% of our genome with chimps. The genetic differences between human groups are real, they cluster geographically, but they're biologically quite small. It really drives home that line from the help. So we's the same, just a different color. The differences are often amplified by culture and history, not deep biology.
But identity is complex.
Okay.
Genes aren't the whole story, but they're powerful. The book mentions David Reimer's story.
Such a tragic story. Born genetically male, raised as a girl after a terrible accident during circumcision, it starkly showed how deeply our biological sex, rooted in genes like SRY, influences our sense of identity, even against intense social conditioning.
And then there was the research into a possible gay gene.
Right, Dean Hamer's research in the 90s, mapping a region on the X chromosome linked to male sexual orientation. It challenged the idea that sexual preference was purely acquired, suggesting a genetic influence. It reopened the door to thinking about how genes might affect all sorts of behaviors and temperaments. Where do you draw the line between nature and nurture?
The Minnesota twin studies seemed to push that line further towards nature, didn't they? The stories are wild.
They really are. Identical twins, separated at birth, raised apart, yet showing these incredibly specific shared behaviors, like the two women arriving on separate planes, both wearing 7 rings, or the guys raised in different countries who both compulsively flushed toilets twice and carried wads of tissues. It's uncanny.
So weird.
And these studies, combined with finding genes like D4DR, which is linked to novelty seeking, really strengthen the case for genetic influence on personality, that D4DR variant. It's associated with both intense creativity and attention deficit disorder. Mukherjee suggests maybe that restless, anxious gene is part of what drove human migration and maybe explains some of our restless, anxious modernity.
Okay, but here's the thing. If genes are so influential, how do you explain identical twins, same genes, raised in the same home, who still end up with different lives, different personalities? What's going on there?
That is the crucial question, and the answer seems to lie in natural ambiguity at the cellular level. Tiny random fluctuations, even in just a single molecule inside a cell, can set cells off on slightly different paths. So even with identical genes and seemingly identical environments, pure chance, these stochastic events at the micro level can lead to different outcomes, different fates for genetically identical individuals.
So chance plays a role right down at the molecular level.
Exactly. It's this idea that our genes provide a script, right? A set of possibilities, tendencies. But we are fundamentally built to go off script. Those random fluctuations interacting with everything else create the unique self. It's this incredible interplay between our genetic blueprint and pure chance.
It's a beautiful idea. Okay, let's look ahead. Tomorrow's code. Genetic intervention is here, but it's had a rocky start. Early gene therapy. Jesse Gelsinger's tragic death. It was a case of genes, but no therapy, as the book puts it. The desire was there, but the tools weren't safe or effective enough yet.
But now we have CRISPR-Cas9, Jennifer Doudna and Emmanuel Charpentier figured out how to use this bacterial defense system to make really precise cuts in DNA. It's like having a molecular scalpel. It's revolutionary.
That precision, though, comes with enormous responsibility. We saw that with the Chinese experiment in 2015, right? Using CRISPR on human embryos.
Yes, and it went wrong. It caused unintentional mutations elsewhere in the genome, hitting essential genes. It was a huge red flag, a stark reminder of the risks when you start editing the fundamental instructions for life. The genome is full of places where we could potentially intervene, these nodes of intervention, but it forces that critical question. Can we make our genomes a little better without risking making ourselves substantially worse?
And projects like ENCODE are mapping even more of the genome's complexity.
Exactly. In code, the encyclopedia of DNA elements isn't just mapping genes, but all the functional bits, the switches, the regulatory regions. It gives us even more knowledge and potentially more power to meddle.
It's almost sci-fi. Thinking about a future where you could sequence, say, 100,000 kids at birth and track their lives to build a fate map, a human phenom, predicting health, maybe even behavior.
It's staggering to contemplate. Such a fate map could offer incredible predictive power, especially for health, maybe even for aptitudes. But the ethical minefield is immense. Who defines normal? Who decides what needs fixing? Who gets to intervene in someone else's genetic destiny? It's a future packed with potential, yes, but also with incredibly profound questions we need to grapple with.
Wow. We've really covered a lot today, journeying through the history, the science, the ethics of the gene, guided by Mukherjee's incredible book. Understanding this stuff, it really does change how you think about being human.
Absolutely. And maybe a final thought for everyone listening. How much of your own genetic story or resume can you actually read or understand right now? And maybe more importantly, when, if ever, do we as individuals, as a society, have the right to step in and rewrite someone else's genetic destiny?
Heavy questions to ponder. We really hope this exploration of the gene has sparked your curiosity and given you a lot to think about. And remember, if you enjoy this kind of thoughtful exploration, do check out the Mindful Moments YouTube channel. If you want to dive deeper into the book itself, we've put a purchase link in the description for you. Please subscribe if you want to keep taking these deep dives with us. Share this with a friend who loves to learn. And always, always remember to think critically about the stories written in our code. We'll be back soon with more podcasts like this one, ready to explore more fascinating topics with you.